All right, so for tonight, we're going to try and do the best we can to make sure that anything that's not making sense, we spend the precious time we have on that. Tuesday night, so Thursday night test, 1 week from today on Module 2. Tuesday night, we will start Module 3. So for Module 3, it's a lot of stuff. It's all immunology. We cover basically everything in the book. It's here down at the bottom. And I'm trying to think. So when you go to the objectives in the PowerPoints, there's the student version of the PowerPoint, denoted by this S right here. And I can tell you, I am going to show quite a few videos because I find that it really helps for that. But you'll notice that this is somewhat of a didactic PowerPoint. See how I have fill in the blanks? If you guys read through, and of course, yeah, this isn't Blackboard anymore, it's Canvas, right? I'll show you where those videos are that I'm going to show. But even if you just read in your book, right? So, what caused the release of the inflammatory mediators, right? See if you can read and fill in these blanks before class on Tuesday. Now, we're only going to get through inflammation, right? We're not going to get to phagocytosis, so you just got to worry about inflammation, okay? Oh, that's not true. We have 3 hours. We will get through this whole lecture. Yeah. But I don't think there's much. Yeah, there's a little bit to fill in on this. It's not too bad. It's not too bad. It's not too bad. So try the best you can to read through inflammation and phagocytosis and fill in those blanks. Like print out that student version PowerPoint. Like, you know, active learning has been proved over and over again, right? If you guys read beforehand and try and engage with the material before I go over it, you're more likely to remember it. And this is probably the most important thing when it comes to immunology, which is why I cover it first. And I don't think they cover it first in the current textbook you guys are using either. So, you know, skip to the section that covers that, right? It's not going to be the first part of that chapter. Does that make sense to you guys? Yeah, it's innate in immunology, like in the 30s somewhere in your book. So if you want to watch the animations beforehand, or any of the videos that I'm going to show, they're, of course, under the tab that says videos and animations. You'll see there's a lot, right, for the immune system. Now, typically what I also have you guys do is read this web page beforehand. It's a good overview of the immune system. But that one, I think we are going to just push to after the test. I may even have you guys, like, read it as an activity that night after the test. Because there should be an assignment there for that. Let's see. It says like immunology web pages or something. Yeah, immunology web pages is a 2-point assignment. It literally is just read these pages and write 2 things you liked about it, so I know you read it. But we're going to do that in class, so I will know you read it. So as soon as you get done taking your test and you take your little break, you'll have your computers, ha ha ha, so you could sit and read 2 web pages. Right? Huh? Oh, you already looked at it? Yeah, I'm not good at the game. You have to fling them. Yeah, I'm not good. I'm not fast. You basically slingshot them. There's apparently a second level and maybe even a third. Yeah.
So don't worry about that until Thursday when we take the test.
Yeah. Yeah, just that PowerPoint, inflammation and phagocytosis. Please read and try and fill in as much as you can beforehand, right? Before that lecture on Tuesday. Okay, so let's get to today's stuff. So I wanted to do some in-group active learning stuff, but I found with that it's better if you have a pre-assessment. So I thought about having you guys do your connects ahead of time, which a few of you guys had, but then I worry about that because then you won't have it as an assessment after you've studied to see if you actually really know it, right? I didn't want to steal that from you. So I think what we will do is, I know for genetics we still had the 3 transfers left, right? So I am going to tell you guys just a few things about those, and then when we break into our groups, I do want you guys to try and explain it to each other, right? Well, let's see how much you know to begin with. So who's going to be our experts on this? So for gene transfer, there's 3 mechanisms that occur naturally. There's DNA-mediated transformation, transduction, and conjugation. So all are examples of horizontal or lateral gene transfer. Anyone know what vertical gene transfer is? Parents to babies, right? So like with women, we talk about some diseases can be vertically transmitted from mother to child, right? So information as far as DNA goes from mother to child, and that's, for bacteria, that really is just mom, right? So this stuff isn't moving, like you've got a spontaneous mutation and mom passes it on. This is stuff, and probably the best example I can give is imagine 2 cousins, right? So somewhat closely related, but not. And my favorite example, which is a real world example. So most people have heard of E. coli 0157, right? This is a pathogenic strain of E. coli. It's sometimes abbreviated, abbreviation popped out of my head. STEC. The S stands for Shiga toxin E. coli. And so what it is that E. coli, this pathogenic strain of E. coli 0157, has the Shiga toxin that it produces, and that's why it's so pathogenic, is this toxin that it produces in your gut if you were to get it. If anyone had it, trust me, you wouldn't be in this room right now. You'd be in the bathroom of the hospital. It's that bad. And you've heard numerous times of things that have gotten contaminated, right? Spinach at one time from runoff of a nearby dairy where the cows were infected with this pathogenic strain of E. coli. Unfortunately, it's happened numerous times to our food supply. But Shiga toxin, anyone know what other organism has the Shiga toxin? It's spelled SCHIGA. Shiga. You're right. Shigella. Shigella. Another gram-negative bacteria, Shigella, produces the Shiga toxin. Okay, so again, how does a bacteria know how to make a toxin? Where is that information stored? In its DNA, right? So in its DNA, right, it has a code that tells the cell to make that particular toxin, that protein, say. So it's the exact same toxin that Shigella has that this particular strain of E. coli has. Not all E. coli has it, just this one strain. So how did it get that DNA? Right? It got DNA, didn't it? That's how it makes that toxin. Could be any one of on this. You see how I'm talking about cousins, E. coli, right? They're both gram-negative rods, right? They're not all that far from each other in the evolutionary tree, right? And one of them gave his cousin the toxin. How did he do that? Could have been conjugation. Could have been transduction. Could have been transformation. It could have been any one of these three. I don't know if there's any research that's been done out there to try and figure out how it actually happened, but it was probably one of these three ways. So, we could take what we know from these pathways and probably, design an experiment to try and figure out if there's any evidence as to exactly how it got there. Because scientists know quite a bit about these processes. So what do you know about these processes, though? Anyone know anything about any one of these three that they're willing to share with the class? Or do I have to break you into groups to get you to talk to each other? Come on, Kat. I know you know something.
Well, transduction uses like a mechanism to take in
Right.
conjugation, they use the F plus and F minus and basically mate.
That's a start. That's pretty good. Anyone have anything to add? Okay, so transformation, right? What I always tell my students to remember is transformers. Sounds like transformation. Everyone knows about transformers, right? My son is convinced that someone who's driving that Camaro around is Bumblebee. Yeah, you listened to my previous lecture, right? He's still convinced. So, that ability to turn from a car into a robot, that's a huge transformation. And we can talk about pretty substantial transformations for bacteria. I mean, what could have happened is that Shigella died somewhere near an E. coli, right? And its DNA became part of the environment. And the E. coli picked up that DNA, found a place because, they're not all that far from each other, right? They're not all that different relationship-wise, which means that, they probably have a lot of the same DNA, right? So there might have been flanking sequences associated with that toxin that matched up in the chromosome for E. coli, right? So he found a spot where it was like, oh, this looks similar to me. Let's plug it in here and see what I get, right? And he incorporated it into his chromosome, and now he had this ability to make this toxin, which ended up being very beneficial, so he kept it. Still with me? So with transformation, they're picking up DNA from the environment. They're going to take it in, they're going to digest it in a particular way, and it's going to be single-stranded. And this is a true possibility, right? Because remember I said, they got to find a place in the chromosome to put it. So only, organisms that are related to each other, per se, are going to find a spot to put it, right? That they're going to have some matching DNA sequences and possibly switch out one gene for another or find a place to insert it in. Now, under what type of conditions do you think that E. coli would be willing to take in DNA? Because there's no guarantee it's good stuff, right? When there's not enough nutrients, that would be a stressor, right? Just like you guys, coming test time, if somebody tempted you with the answers to the test, you'd be like, tempted, would you not, right? You can't help it, right? You're stressed, right? This would give you an advantage. And so when they're under stressful conditions, crowding, lack of food, lack of any necessary component for life, then they're more apt to become what's referred to as competent. And only competent cells will actually take in DNA. And so in the lab, we can actually cause this to happen, right? Those of you guys that are taking the lab later on in the semester, you'll actually work with a strain of cells of E. coli that have been made competent. And we're going to stress them even more and put them under conditions Well, they'll actually take in a whole circle piece of DNA. They're not even going to incorporate it into their chromosome. They're going to suck it right into their cell, and it's going to stay intact as a circle, as a plasmid, as an extra circle of DNA. And it has a whole bunch of extra information on it that we're giving them. And because of that, they're going to transform. They're going to completely change and be able to do an ability they never were able to do before. They're going to become resistant to ampicillin, because on that DNA plasmid we have an ampicillin-resistant gene that makes an enzyme that destroys ampicillin. And we're going to give them the ability to make green fluorescent protein, but we're going to put it under control. Right? So they're only going to do it when the sugar is present. Ooh, one more. Good evening. And green fluorescent protein does just that. You shine UV light on it, it fluoresces. So, I mean, that's a big transformation, is it not, for E. coli? So, again, it's possible that this is what happened with the whole Shiga toxin. He picked it up from a dead Shigella, found a place to put it in the chromosome even, and utilized it and was able to produce this toxin now. Kat was right. In transduction, you need some type of vehicle. And that's why I always tell my students, too, to focus. Because, you know, we just said transformation, didn't we? We said trans. And now we're saying trans again. And some of us kind of just stop there, right? But the rest of the word is different. It's transduction. And really focusing on that duct word. Because when we say duct, we're referring to traveling through something, right? So most of the things, you know, water travels through ducts, right? And in this case, the DNA, remember all the time, this is what we're talking about moving is DNA, right? The DNA is going to move from one bacterial cell to another. The duct, or the vehicle in this case, is a bacterial virus, commonly referred to as bacterial phage, or just phage for short. You want to talk about play on words? The conference that I went to, some of the people that were in that one are in another program called sea phages, and I thought they meant like the letter C. No, it's like sea, S-E-A, as in the ocean, right? Yeah. So apparently they're studying viruses of the ocean, which is quite fascinating because, oh really, you're going to call me now? It ain't happening. It's my husband's doctor's office. Why are they calling me? It's 6:30. All right. They want $100 to fill it out. Okay. Okay. So duct, right? Is a virus. Okay? So, all right. We know about viruses. We did that before genetics, right? So, what happens? How does a virus infect a bacteria? Right. They attach and then they inject their DNA into the cell. And then that's it? Right, they can go lysogenic or lytic, but depending on whether it's a temperate phage or not, right? But say it's lytic cycle. So they're going to bust out of those cells, right? First, once they're inside the cell, they got to get that cell to what? Not be a cell anymore, right? Be a virus-making factory, right? So they're going to get that cell to copy their DNA, get that cell to make their proteins, right? Get them to assemble, and then they're going to bust out of that cell and then go infect another cell. Does it always do it perfectly? No. And one of the things they do to stop the cell from being a cell is they have enzymes that will chop up the bacteria cell's DNA. So it can't use it. The problem is that apparently sometimes instead of packaging their viral DNA inside that protein coat, they pick up some of those pieces of the bacteria. So now when the virus pops out, it doesn't know it has the wrong stuff, right? Its outside components are going to target a particular cell. And again, it's going to be within particular groups of cells, right? That sometimes they're going to attack. So that one's going to go land on another bacteria, and it's going to inject the DNA inside. But is that bacteria going to make more viruses? No, it just got injected with its buddy who died, a couple blocks away. DNA. So it just got a, little injection of a present, potentially, right? And again, that cell might just degrade that DNA that was introduced, or it might incorporate it into its genome. So that could have been a way that toxin traveled from Shigella to E. coli. And there's some things, that happen with those mechanisms when you really get down to the molecular level that they could probably potentially trace, see if that was the possibility. They've actually found, and I think this is why this group is studying C phages as in SEA instead of the letter C, is that I read a paper several years ago, and they found that the pigments in algae that are in the oceans, that ability to make those different pigments that they do to allow them to photosynthesize, they believe that they're acquiring them from one another from viral infections. So a virus that infects algae is picking up DNA on how to make the pigments to photosynthesize and is transferring it to other algae when it tries to infect them. So it's messing up like what happens with bacteria with transduction. Make sense? Kind of cool. Then the last is most people's favorite is conjugation, because most of you have seen that in other organisms. And so sometimes it's given the alternative name of being bacterial sex, right? Because all sex is, well, not all it is, but the main part of sex is exchange of genetic information, right? In scientific terms, that's what makes it sex, or sexual reproduction, right? As opposed to asexual, where the exchange of DNA does not happen, right? You just copy the stuff you've already got. So with this, they literally are joining up by the sex papillus, or the papillae, or the fertility papillus. And one bacteria is transferring DNA through that protein tube to the other one. So this is the only form of these 3 forms of gene transfer where the donor, where the DNA came from, actually survives the process. Just think about it. What did I say happened for transformation to that Shigella that donated probably the DNA? It died, and its DNA became part of the environment. What happened to the virus, the donor, where the DNA came from for transduction? It died from what? A viral infection, a phage infection. This one, the donor actually survives, right? And these and a lot of times they'll transfer plasmids, but they can transfer chromosomal information too, but I usually keep it to plasmid because that's the least complicated of the conjugations. A lot of times on that plasmid, on that circular piece of DNA, is the information on how to make the sex papillus. In addition to, other genes that give them the other abilities and why they want to share. So the really frightening thing about the antibiotic-resistant world that we live in, there's lots of plasmids out there, right? So these smaller circular pieces of DNA that contain information on them, like say an enzyme to destroy ampicillin, right? So they contain information on how to be resistant to antibiotics. So we give these plasmids a special name. We call them resistance plasmids or R plasmids. And the terrifying thing about this is them sharing this with each other, right? You stick one E. coli in a test tube with a bunch of his buddies who don't have the plasmid he's got, he'll give it to all his friends. And the thing is, it's like a sexually transmitted disease too, because I mean, again, it's like sex, right? They're transferring information. They're transferring DNA. Once he gives it to one of his friends, that friend can then give it to other people, right? So like they say in sex ed class, it isn't really who you slept with, but it also is who that person slept with before you, right? That's also the problem. So you're not just concerned about one person, you're concerned about all the other people potentially in their line of interactions. So that's why, too, it's going to spread like wildfire in that test tube of E. coli, because he gives it to one, now that other one has it, and then they go give it to their friends, and it just goes exponentially. I stayed in a dorm for college, so we were required to go sex ed, at least one sex ed workshop. Right? I think every, probably middle school student at this point should be required to have those classes. All right. One funny story for you guys. So, we're in this big lounge area, and the guy who taught these classes, this man, I mean, he prided himself on that no one could ever embarrass him ever, and I really truly believe this for this man. He was impervious to anything sexual related, and so he was talking about several different diseases, and I can't remember which one this was, but he was talking about how for males a crust would form over the tip of the penis. So because of that, when you go to urinate, you'd get what's called split stream, right? Where it would, the crust would cause it to not be one stream, but split in different directions. And then he prefaced it with, well, sometimes that can happen if, say, semen dries at the tip of the penis instead of, this infection. You could get the split stream. So there was a poor guy in the back, way in the back, sitting, I think, in the windowsill. Let out the biggest sigh of relief you've ever heard in your life. Because apparently he had experienced the split stream, and he was terrified that he had this sexually transmitted disease, but then figured out probably it was the whole semen at the tip of the penis problem. Right? So, yeah. Oh, it was loud. 'Cause, I mean, it was quiet. And so, yeah. Is interesting. But yeah, I think that too, again, you got to kind of tell yourself stories with these things, right? To help separate out these terms so that you can keep them straight in your head. So you guys feel a little bit better about these three forms of lateral or horizontal gene transfer. Okay. So I'm not going to hash those out anymore. At this point, I'm not going to make you guys talk about them unless you want to. And I'm not going to look at this lecture anymore. We're done. That's just specifics. We got the gist. So let's look at, growth. In terms and media. All right, we'll jump around a little bit on this one. I'm trying to think. Oh, okay. Before we go to break, I'm going to jump ahead. And those of you guys in lab, this stuff is coming up. You actually inoculated this week. All right, so lots of terms, unfortunately, in this second component. I'll tell you right now, as far as the temperature, I just want you to know, like, what's the lowest to the highest in order. I don't want you memorizing any of these ranges per se, right? Just having a general understanding of these environments. So we'll look at that for a moment. A few numbers you do really need to know range-wise, though, are pH, right? So for acidophiles, you need to know that their pH range is 0 to 5.5. Neutrophiles 5.5 to 8, and alkalinophiles from 8 to 11.5. And then, of course, the oxygen requirements, you really do need to understand those definitions. So what I do with this component is that we talk about these influences, you know, the availability of water, pH, which anyone know what pH is actually the measure of? Acid base, but something is acid or basic depending on in the solution how many hydrogen ions are present. It's actually the little p stands for the partial pressure of hydrogen ions, or also referred to as protons, because the hydrogen atom is just a proton and electron, and usually gives up its electron a lot of times, so then it's just a proton. Right? So, do you have too many? Are you acidic? Do you not have enough and you're alkaline or basic? Or are you balanced at neutrality? And anyone who's taken anatomy and physiology too knows acid and base is very important in the blood of our bodies, right? It's a very narrow range, 7.35 to 7.45. You go below or above that range, you experience some really serious stuff and can potentially die if it's not fixed soon, right? You can go into acidosis or alkalinosis. One of them, you stop breathing, go and slip into a coma and die. One of them coma and die, and the other one is convulsing, can't breathe and die. I can't remember which is which. I don't have to though, because I don't ever plan on, doing that. But this is why when people take things like aspirin, which is an acidic drug, you could die because your blood gets too acidic and you go off the reservation. So, not good. But also, like, your breathing rate can affect your acid-base balance too. Your breathing rate can help with the acid-base problem, or it can cause an acid-base problem, right? And then your kidneys are all the time trying to fix an imbalance, right? Because they're going to throw any excess acid into your urine or any excess alkaline into your urine. That's why your urine can fluctuate quite a bit acid-base wise, because that's your get the heck out of here, I don't need you right now. So for the most part, microorganisms are going to maintain a neutral pH inside, but some of them have adjusted and adapted to where the outside pH can be high or low and they'll be okay, right? And that's when they'd be classified as either acidophile or alkalinophile. In other types of organisms are more resistant to acidic conditions too, like fungi, right? Can deal with acid conditions a little bit better than most bacteria. We all know temperature affects growth, right? That's kind of a no-brainer. We use that all the time to control microbial growth, right? And that's why this lecture, I have it as control and growth. Why separate these things? What they need to grow, because we know they need it, we turn around and adjust that one way or another to control the growth of microorganisms. Oxygen concentration, pressure, radiation, right? All of these can affect growth, and then we can apply that information to control them. So water's really important. No living organism can survive without it. So if you're in a situation where water is less accessible because there's a lot of solutes, that's going to create a serious problem for you because you've got to find a way to compete with those solutes, which are like, no, 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 it's my water, you can't have it. And you're like, oh, no, no, I need that, right? Let's make a deal, right, about the water because I need it, right? So some organisms have actually adapted. And what type of environments on Earth is water actually really hard to get because solutes are like trying to hog it? So what's a solute that usually creates water problems? What's something you could put in water that's going to want to keep the water to itself? It would create what we call a hypertonic solution. Salt. Sugar can do it too, right? And so if it's really, that water's really salty, it's less likely that the water's going to go into the cell. You actually probably have more water inside a cell than you do in salt water. So those cells are fighting the problem of, I need some more water, but the surrounding environment doesn't want to give it up. So they adapt, right? And sometimes they adapt so much to one extreme that if you take them out of a salty environment, they'll die, right? Because they've adapted so much to that environment that when there isn't any salt, they can't balance themselves. So some organisms are osmotolerant, right? They can do this problem, they can tolerate it within wider ranges than most organisms, right? They've developed compatibility strategies, right? Special molecules that help them combat this problem. Bacterial cells, not many of them are so good at this, right? They'll undergo what's referred to as plasmolysis, and I hate this term. And one of the reasons why I hate this term is because it sounds like the cells pop, right? They don't. What do? They shrink, right? They shrink. And so how do we create hypertonic solution to preserve our food all the time? High salt. And honey is a hypertonic solution. It's not salt in that case, it's what? Sugar. High concentration of sugar, right? And that's why, for the most part, you can leave it out, right? And you don't have to worry about bacteria growing in it. What do you have to worry about growing in it, though? Not bacteria. What are other microscopic organisms that sometimes create problems for us? Fungi, right? And remember I said earlier, they do better with acid-base. They do better with hypertonic solutions as well. Right? So they always tell you not to give it to babies, right? Because there's the possibility they could have a reaction that something may be contaminated. Then there's the whole problem with pollen too for some people, right? Because that's how it came to be, by the bees. So, is this a permanent condition? Like if you salt something, but then later you soak it in a whole bunch of water, will it pop back? Yeah. And so if it's a bacterial cell, right? So you shrunk it up, you made it really unhappy, it wasn't really growing, but you didn't actually kill it. So that cured ham that you then stick in the water and cook, if it's contaminated with an organism, you might just revive it by doing that. Right? So Staph aureus, all the staph epi, are salt-tolerant organisms to begin with. So salting doesn't usually stop them. So you're like, oh shit, right? I thought that cured ham was going to be fine, right? Isn't that the way we preserve ham, and it's going to be fine? Right? There's no one perfect way to preserve food, for one. We do the best we can. But it's good to know about these things so that you can look for them. But that's also why, too, if you have cured ham, go ahead and cook it, right? Won't hurt to hit it with another problem, right? So salt was one problem. Let's try how you do with heat, right? Let's see how you handle that one, right? They're not usually going to survive multiple methods, right? And heat is probably our most common. Another term you'll sometimes hear as far as osmotic pressure is halophiles. Halophiles are those ones, that example, like I said, where they've gone to the extreme, where they've adapted to salt, right? Halo referring to salt, so much so that they can't survive without it. And we're talking, some of them have even become extremes at this, extremophiles, where they need 15 to 30% salt. Where on earth is that? Great Salt Lake, right? And the Dead Sea, right? Places like that. The ocean isn't even that salty, y'all, right? These are like serious salts. So agar, right, which we use to solidify our media for our plates in some of our slant tube media. We can't put this in too high of a concentration because it's a sugar, right? It's a sugar for marine algae. So would it affect osmotic pressure? Yeah. So typically, you don't usually go above 15% agar in most of the stuff that we use. Because anything above that, it's gonna start pulling water out of the cells you're trying to grow, right? It's too, quote unquote, sugary, I guess you could say. Even though we haven't found a single microorganism that can digest this sugar yet, I'm probably sure there's one out there. We just haven't found it yet, right? There are bacteria that figured out how to use oil as a food source, right? Like crude oil, right? So I'm sure there's probably one out there that eats agar, but we haven't found it yet. But yeah, this does affect osmotic pressure. So we do have to watch the concentration that we put in with the food, in with the media, when we grow microorganisms. So pH I mentioned, right? It's actually the inverse log of the hydrogen ion concentration. And we use acid. You probably use acid base a lot. So I didn't get a chance to look at your guys' chemicals used to clean, but probably a lot of you guys are using acid-base imbalance to disinfect, to kill microorganisms or inhibit them. So you'll notice on this list is good old bleach, right? And so again, most organisms are going to want to stay in a relatively neutral pH. So when you throw bleach at them, that's very basic. That's more than they can handle and it kills them. When you use the other end of the spectrum, say use something like vinegar, which a lot of people use as a nice natural cleaner, right? You're going to acidic on them. Should you mix these two? Certainly not. And which two cleaners they always tell you not to mix and must always inform you if it is in the cleaning agent not to do this? Ammonia and bleach. And that is an acid and base, but worse than that, worse than canceling each other out, apparently it produces a toxic green gas that you don't want to inhale. And with bleach in general, you don't usually want to inhale it either. You want to in a relatively open space. I feel the same way about ammonia. I never use ammonia. It smells bad to me, right? Some people it does because we do sometimes have a certain component of ammonia in our urine. So for some of us, no matter what, I don't know, ammonia always smells like urine to me. I can't overcome the smell. Not a big fan of bleach either. I use other alkaline cleaners like Lysol for the most part. But yeah, that's why something like lemon juice too, when you put on fish to make ceviche, lemon or lime juice, those are acidic, right? All your citruses are acidic. It has citric acid, right, which is vitamin C. It's also what they put on all that sour candy, right? It's just vitamin C in high concentrations, citric acid. Acid is sour tasting to us. Right? I'm developing a better taste for vinegar. I used to just not even be able to at all eat or have anything with vinegar. So I'm slowly adapting to the southern lifestyle because, they put that on just about everything. That and, a lot of spice. A lot of spice. Keeps my nose running. It'll help you when you get stopped up. But, that's the reason for some of this stuff, too. I mean, and that's why you see, it's a chemical change. Like, once you put it on the fish, it changes in texture, in color, right? There's a chemical reaction that's happening there. And again, one of the main reasons why they did that wasn't to change the texture or the color of the fish. It was to kill any microorganisms that may be on that fish, right? And then we, got so that we like it that way. So acid-base balance, very important for most organisms. Some of them can handle acids. So if you're using vinegar to clean, it might not be enough, right? You may have to use bleach instead, right? And go to the other end of the extreme. But you never want to mix an acid with a base because then they're going to cancel each other out. You're going to get water and a salt, right? And you just defeated the purpose. Some people that are super crazy cleaners, one of the things that you can do to really make sure you're kind of getting everything is switch between two different cleaners. One week use one, the other week use a different one. And so therefore you're changing the possible populations that might be there. And bettering your efforts. In the food industry, they use bleach, right? But is it just plain old bleach in water? No, it's usually hot water in bleach, right? If they're really serious about cleaning. So we're adding another element. We're not just dealing with pH, we're also using heat. Make sense? So whatever makes them happy, we try to go the other way. All right. So we use it a lot in cleaning, but we also use it in food preservation. Can anyone think of foods that are stored at extreme pHs? Acidic is the most common. So another reason why I don't really care for pickles. Right. So a lot of them, depending on the process, they might be fermented, right? And that produces acid, which impedes other microbes from growing. Sometimes they just put them in vinegar, right? Like pickles in vinegar. You've got sauerkraut, some kimchi. Yep, I'm not a kimchi eater. I like the pickled, the cucumbers done kimchi style, but not the cabbage. That's traditional kimchi. But then the spices and everything else, right? Vietnamese food. But a lot of cultures have a lot of fermented foods, right? And this is acidic, right? Most of the time when you have fermentation, you have acid production. And it's that acid that helps preserve the food because it helps stop the growth of microbes that don't like acid. Does this mean that it completely preserves it? No, right? You have the possibility of an acidophile that's like, oh, this is great. Make sense? Okay. So temperature. Every organism has their optimum best temperature, right? For humans, it's around 37 degrees Celsius, which is 98.6 degrees Fahrenheit. You start going too higher than that, right? Growth is going to significantly drop off and can even lead to death. What molecules are most affected by heat in an organism? What do you guys think? So who are the workhorses in our cells? Who catalyzes all the reactions and the chemical reactions? Enzymes. What type of molecules are enzymes, though? Are they lipids? Are they sugars? Are they nucleic acid? Are they proteins? Which one? They're proteins. They're proteins. And what happens to proteins when you heat them? They denature. They change form, and they can change form permanently. And is form related to function? Oh yeah, right? When you think about enzymes, they always draw these little Pac-Man things, right? With the active site, and it grabs onto the substrate, and it does something with it. Well, if you deform and now become like this flat pancake, can you still do your job? No. So when we heat them up, right, we affect those bonds, we change them, and we change them permanently. So the best thing, other than, this is your brain and this is your brain on drugs, but the whole fried egg thing, right, frying an egg, once you fry that egg, right, that's it. You can't get it to go back to the raw egg, right? Because you permanently change those proteins. They're stuck like that. And also, if it's a non-stick frying pan, they're really going to stick. Because proteins become really sticky when you heat them. Like glue. Right? So, when you heat stuff up, right, this is why you can cause death with heating. When you cool stuff down, though, do you kill it? Depends on the organism, right? When it comes to microorganisms like bacteria, pretty much just slow them down. That's what we do when we refrigerate and we freeze. And freeze, you can put them in like stasis, right? Animated stuck. And then when you take them out, they go, hey again, I'm still here. Little evil buggers. It's kind of good for me, because, we just store our microbes in the refrigerator and the freezer long term. We just wake them back up. It's not so good for your food that's sitting in the freezer, right? Depending on how much was there to begin with, and when you take it back out, it's probably still there. So you're going to do what? You're going to cook it. Chances are you're going to cook it. And you got to cook it all the way to make sure you kill them all. And so that's the reason, too, is like, you're like, well, why are we cooking chicken different than beef and different than pork? Why all the different temperatures? Why not just one temperature? What do you think, Ashley? Yeah. Exactly. So the most common offenders for, say, pork, we know those microbes, and we know how hot you've got to get rid of most of those bad guys. And the ones commonly associated with beef, we know, and so we know in general about how hot you've got to get, that you're going to kill most of them, and yet not, burn the crap out of the steak you want to eat. And then let's I don't know anybody, I know a few people that actually eat well done steak, right? Because it's just really not good, well done. Might as well just chew on some leather. But, right, other things like ground meat, that's a slice of meat, steak. And I know Kat's dying right now. But, but when you grind stuff up, different parts from different areas and stuff, you don't really exactly know all what's there, right? So the likelihood of contamination increases. And this is why when it comes to ground meat, they say cook it all the way, right? All the way through. No pink, no red, no nothing, right? Where, you can risk it if you know that, it was a good size one piece of meat that they ground up and made into that, or if you ground it yourself, then yeah, you could cook it medium or something like that, right? It's when you've got these lower grades that may have been mixed in there and there's the possibility of contamination. That won't make Kat throw up. But a friend of mine, I don't know why she would even order hamburgers, because we'd go out to eat and she'd tell them, cut it in half, make sure it's cooked all the way before you bring it out, because if it's not, I'm going to send it back. And the waiter's like, okay. I said, no, honey, you don't understand. Do what she said. Don't bring it out here without cutting it in half and looking at it before you bring it to the table, otherwise she's going to send you back, and I'm not kidding. You're just going to make this longer. And I'm hungry. But, she had a bad experience, and you can't blame her, right? I just personally would probably skip getting a hamburger when I went out to eat. But that's just me. But you understand the reasoning behind this now, right? So you can explain it to your friends and your family even, right? The reason for those numbers, right? It is really still our best guess at all of this. Or you can just do like Kat does and just avoid the whole problem to begin with. Except Kat, don't forget, right? They contaminate sometimes your beautiful vegetables with animal products like runoff from nearby dairy farms. So it's not a 100% either. So we do the best we can. But, you know, most of us, like I said, we use this all the time, but do you really think about why it is we refrigerate things, right? Right? It was for preservation, but it's preservation because microorganisms are in our environment. You can't avoid them. They're on your food. They're in your food. And so the longer you have it, the more they're going to grow, and the less likely you are going to want to eat it. Still with me? All right. So now I'm going fast. So these temperature ranges. So each organism has its favorite little area, and these are based on whether your optimum falls into that area. So it doesn't mean if it's a mesophile that their optimum temperature is 37. It means it's within that range of around 37. Make sense? So meso's like in the middle. That's us, right? And then you're way down here, it's psychophiles. And psycho means cold, by the way. And I feel the same way about things that like cold, like this cold. They're crazy. Right? So psychophiles is the bottom, the coldest, right? So we're talking, and again, this is Celsius scale. So we get kind of like messed up, right? Because most of us don't deal in Celsius. Celsius is based on the properties of water. Water does what at 0 degrees Celsius? Freezes, which is what in Fahrenheit? 32, right? What happens to water at 100 degrees Celsius? It boils, which is 212 degrees Fahrenheit. So we're talking about not frozen water, right? Because all living organisms need water and not in the frozen state. Right? It's got to be in the liquid state. But it can be really cold liquid state, right? And they can deal with that, right? So your refrigerator is usually around 4 degrees Celsius, right? Which I think is like 20 degrees Fahrenheit or so, something like that. Room temperature, if I had it my way, would be 70. Although I have to say, living in the South as long as I have, I think I actually would prefer like 74. 70 feels cold to me now, or it didn't used to. And then good old body temperature is 37, right? And notice that's the mesophiles, that would be us. And just to give you an idea of how hot 70 degrees Celsius is, it's 158 degrees Fahrenheit. Pretty darn hot. So what type of organisms are we talking, what type of environment should we say are those psychophiles in? Like Antarctica, right? In Alaska, right? In glacial lakes, you know, I mean, where it's just barely water, right? Do we have to worry about any of them infecting us? Like if we went and got a fish and ate it out of one of those glacial lakes? Probably not. Why not? We're too hot. We are really too hot for those organisms that would be associated with that fish. Now, psychotrophs, on the other hand, do we have to worry about some of those? Yeah, and some of these guys are what will continue to grow in your refrigerator. So you put in the fridge, and it's like, ooh, cool. I'm all about this. And one of them that could kind of cross the line, because notice there's some close crossing there, right? Is listeria. All right, listeria can grow in us and in your refrigerator. Right? This is why pregnant women are advised to avoid certain foods that are commonly contain listeria, because not really so much for women, but the baby, it can cause serious problems. And one of the case studies, if you guys did it right, was on the woman with the listeria, and the baby was born with meningitis. Survived, thank goodness, but had it been earlier on in her pregnancy with the whole hot dog debacle, then she probably would not have had her child. Right? So, yeah, again, we know, the thankful thing about the time that we live in, we know about these things and we can avoid them or You know, make sure you cook that hot dog all the way. You don't contaminate it with anything else, right? And I love the cooking shows they have nowadays, like Rachel Ray, and they're really good about telling people, I'm washing my hands right now because I just touched raw chicken and now I got to go do something else, and I don't want to transfer any of the microbes that are inherently present on these food items, right? We don't want to cross-contaminate. You don't want to use the same cutting board that you just used to cut raw chicken to cut up your vegetables that you're making a salad out of and you're not going to cook, right? So you're not going to kill those organisms. And in Louisiana, turtles, right? They actually have on the books in legislation certain size baby turtles that are allowed to be sold in pet stores. Supposedly, they're large enough that a kid can't stick them in their mouths. In general, they carry things like salmonella and it doesn't bother them, right? They don't get sick from it. So if anything, it should come with a caution to parents. Do not let your children play with this unsupervised, and always make sure they wash their hands afterwards. You know, but most Louisiana Cajun boys and girls, not to exclude the girls, know things like this. Like alligator snapping turtles will take your finger off too. Mesophiles would be us. So again, anything that infects us, right, usually falls into that category because they're living inside of us, so that's going to be their optimum. If it's not, they wouldn't be there, right? They wouldn't survive. Thermophiles, right, are things in like the hot springs in Yellowstone National Park and compost heap. We really have to worry about them making us sick? No. And the problem too with those, some of those refrigerator guys, other than Listeria, some of them will grow at refrigerator temperatures, but they wouldn't grow in us. The problem is the toxins that sometimes they'll produce and that are in your food, and then you ingest that and it makes you sick. So that's another thing to take into consideration. But as far as we know, there's nothing that grows in these really hot places that would actually grow in us, right? Hot tubs, remember, are not as hot as you think they are, too. If they were like the range of thermophiles, you would be dead, right? We really can't get too hot, right? The whole denature the proteins, trash down. And we'll talk about fever when we get to immunity next. Hyperthermophiles, so these would be down in the hydrothermal vents in the ocean, right? Where like magma from the center of the earth is bubbling up into the ocean. They've actually found microorganisms, y'all, growing at that extreme heat. I mean, just, you know, that much away from boiling. Like, look, right on the edge. Actually, probably, like, almost, like, that hot. Crazy hot. Crazy, crazy hot. PCR nowadays, polymerase chain reaction, if you work in genetics or related to DNA or RNA, we actually have thermophiles to thank for that. Because the enzymes that they use to copy their DNA, we've stolen, and we put them in our little test tubes. And when we heat up the test tube to separate the two strands of DNA, the enzyme does not denature. It keeps working. So then when we cool it down, right, enzyme attaches, copies the other strand of DNA. Then we heat it up again and separate the pieces of DNA. Enzyme goes at it again when you cool it back down. It keeps going and keeps going. Old-time PCR, they'd run a reaction, separate the pieces, add more enzyme. Do it again. Add more enzyme, because each time they heated it up to separate the pieces of DNA, the enzyme would denature. So they had to put fresh enzyme in. So really cool organisms like that allow us to copy DNA in a test tube. Using a machine, right, that goes hot and cold, hot and cold, hot and cold, over and over again, cycle after cycle. You just put it all in the test tube, stick it in the machine, walk away, come back. They have ones that are like this big now. You can hook them up to your cell phone or your computer. It's crazy. High school's doing PCR. So this is actually a picture in Yellowstone National Park that I took when I went there. and it's a pretty amazing geological situation. So chambers of that have a particular architecture to them, and water that's being heated, and then it just shoots out. And there hasn't been any earthquakes or anything that have changed the chamber where the water heats up for Old Faithful, and that's why that geyser pretty much goes off like clockwork. Mammoth Falls, which is another thing that you'll see in the old-time movies and stuff, where there was this water that came down over these different terraces. It doesn't flow anymore because an earthquake happened and shifted, and the water doesn't get there anymore, which is kind of sad. So it's constantly changing, this place. It's constantly changing. It's a big caldera left from a volcano that exploded. God only knows how many billions a year. I don't even know. I can't even guesstimate. So there's really, again, it's, water traveling through these different areas, and there's the volcano still down there heating all this stuff up. But one of the fascinating things, and this was the only one that I could really kind of get it all in one shot, you see the different colors? Why do you think you see those different colors? Different temperatures, right? Because it's really hot closest to the magma, and then as it gets out, it's cooler. And so we already know different organisms live at different temperature ranges, right? In this case, they also happen to be different colors, which is pretty cool. There's one that they call, rainbow something, and it's enormous. It has all the different colors. It's just beautiful. And when you go there, I would be like, oh, this one's my favorite. Don't say that. Because 5 seconds later, you'll be like, oh, this one's my favorite. My ex-husband was like, I was like, yeah, I know. Just when you think you've seen the most amazing thing there, next section's going to top it, for sure. You'll see more bison than you have ever saw in your life, unless you've been exposed to them previously. You will actually stop taking pictures of them. I did. Hopefully you'll be smart and don't do like stupid people. Everyone's pulled over on the side of the road. We pull over. Hey, what's over there? A bear. Okay. We drove down the road. Everyone else is heading towards the bear. It is like a drive-through safari, though. I mean, there's like animals everywhere, and they decide where they're going to go, not the other way around. There was a buffalo just taking his sweet time crossing the road. They're like, okay, anytime now. I'm hungry. I might eat you next. I did too. Not that one, but a different one. I love bison. It tastes really good. Cat's like, shut up now. Okay, so amazing place if you ever get a chance to go. On all scientific levels. And just beautiful. So how is it, because we already just said, right, you're a protein, you heat up, you go, right? So how is it that these guys don't go splat when they're in these environments? How are they living? Well, obviously they have adapted. Remember when we talked about, did we talk about saturated, unsaturated fatty acids? We did in membranes, right? And also archaea can do what with their membranes? They can shrink, right? They can cyclic. They can even have monolayers, their membranes, where, I mean, think about it. If you're hot and you're a single layer, you're less likely to come apart than if you're 2 layers, right? So they have more hydrogen because that's saturated. Remember, that's going to need to take a lot of heat to be able to melt it. Where, butter, remember, that's saturated. That's why it's solid at room temperature. But when we heat it, it'll melt. So they've got a lot of hydrogen, so they can resist that melting. Proline is an amino acid that apparently is pretty resistant to, and if you looked at its structure, it kind of makes sense, to heat conditions. Chaperone proteins, right? So what do chaperones do? They watch over, right? So in this case, kind of keep the other ones in line. So they help them out. They can't do it on their own because there's this whole heat problem, right? That temptation of melting. They have histone-like proteins, and these are the proteins that are associated with our DNA, right, that help stabilize our DNA. So they have proteins that are very similar to eukaryotes that help them in stabilizing their DNA. And again, more saturated, more branching, The different linkage we see in archaeal membranes, right, is really one of those adaptations for temperature, where they have that little bit different bonding structure. So we know all about temperature, right? We use it all the time for controlling microorganisms, whether we're freezing them, chilling them, or heating them up and killing them. Right, that one's a no-brainer. Oxygen can be the tricky one. And we might need to take an oxygen break. So let's take a 15-minute break. I'm going to come back and we're going to go over oxygen. And then I'm going to do a little activity with you guys as far as the media goes from the previous lecture.